my jam, you know what I mean. So, really enjoy it. But before I start, I want to make sure I highlight the people that actually do the work. Mr. Ricard is not in the lab anymore. We're lifting data he generated several years back for us. Sean, who you heard from yesterday, one of the speakers, joined the lab recently, the new Amendra, new and improved, we'll say, make you feel good. Bailu and Brad both will have presentations coming up directly soon after mine, so you'll get more information on some of the projects I'll touch on that they're doing, especially Bailu. Brad's independent, but was a postdoc in the lab, so some of the work he did when he was a postdoc. And then Samir Patel and Sasha Wachowski. Samir's here. Sasha wishes he was here, but he's recovered from a surgery he had 3 days ago. So, me and Sasha, I think, first started talking about mitochondrial transplantation back in 2010 over the winter break. You know, it is only professors are in the office on winter break. We were there. We're banging some ideas around it. And mitochondrial transplantation was one that came up. So, when we talk about mitochondrial dysfunction following any kind of CNS traumatic injury, I want to give a brief overview of what it looks like, how does it manifest itself in the mitochondria. Importantly, is it amenable to treatment? I've spent 20 plus years looking at treatments targeted to mitochondria, and we do think it is amenable to treatment. Does it matter for functional outcome? That's why we're all here, right? It doesn't matter if we can save all the mitochondria if we don't have some kind of functional outcome. So just for if you don't study traumatic brain injury, one of the reasons it's so close to near and dear to my heart is that really it's a huge problem. About 1.7 million people in the U.S. Seek medical attention each year for traumatic brain injury. They can break down into falls, motor vehicle accidents, or just being struck by something, right? You know, hit by a car or some kind of injury such as that. I've been studying this a long time, and really the only thing we've done in the last 40 years is get people to the hospital faster, to the ER, so we really need progress in this field to find some kind of intervention. Spinal cord injury, again, just bleak statistics. Really the only intervention we have that's approved at the moment is decompression of the spinal cord injury itself, so that the spinal cord doesn't stay compressed very long. And again, I got a call 3 weeks ago from a cousin who her husband had been feeding, grew up in a cattle region, feeding hay in a row bale of hay, rode back on the tractor and gave him a C4 spinal cord injury. So, you know, it happens all the time, and just the cost of the people is something that's staggering. So again, no real treatment. And part of the reason we don't really understand completely how these progress. So we use animal models to study these. This is a shot of what our controlled cortical impact model looks like. We can impact the brain, get a very controlled type of lesion. So we can look at a mild, moderate, or even a severe type of traumatic brain injury. Same thing with the spinal cord injury. We have a pneumatic piston that drops in the spinal cord, leaving a big cavity in the center of the spinal cord. And what we try to do is spare this tissue or improve the outcome in both these cases so that we can have restoration of neurological function. The reason we take mitochondria involved, just briefly, if you know anything about glutamate excitotoxicity, after a traumatic brain or spinal cord injury, glutamate excitotoxicity goes through the roof. Calcium rushes into your cells through synaptic activity. Mitochondria are huge calcium sinks, if you didn't know that. For each 30 millivolts of membrane potential you have, you take up 10-fold the concentration of calcium that's in the environment around the mitochondria. Huge calcium sinks. If you overload them, you end up destroying the mitochondria. They go through something known as permeability transition. They actually dump all the calcium back up and rupture in some cases. They can't buffer any more calcium, and they actually become net consumers of ATP instead of producers. So it's catastrophic when we go through this type of thing. And we've established this over the years to be very prominent in both the spinal cord as well as traumatic brain injury. And I'm an old school, classic mitochondrial bioenergetics trained person. So we use a lot of oxygen consumption, OCR generations to look at different states of mitochondrial restoration. And the take home of this is that if you look progressively over time, we spent a long time doing time courses, for instance, of how the injury progresses in relationship to mitochondrial function. And what you see is it is progressive over 24 hours. Same type of picture in TBI, traumatic brain injury. We have a big drop, and we have an artificial inflation, we think, of immune cells infiltrating the injury as the blood-brain barrier breaks down. And then that seems to get progressively worse and then just kind of restored. It never gets right back up to sham levels. So we're trying to figure out when to intervene in this. Make sure it's at 5 minutes. I haven't talked that long already, I'm sure. But, and the reason for this, you'll see now, we've noted some interventions, such as using this of FCCP, which is a mitochondrial uncoupler. These are just weak acids that are lipophilic. They'll actually move protons across the inner membrane of the mitochondria and depolarize the mitochondria. And you can use it in a mild iteration so that you don't stop ATP production, but you block the amount of calcium that's taken up, and you also block free radical production. And you can see that if we come in, this is what sham looks like after an injury. And if we administer FCCP beginning at different times post-injury, we can restore a lot of this mitochondrial function that we see drop after a traumatic brain injury. And even seems to be out to even a 24-hour administration, we can actually restore some function. So it's a chronic window of intervention. Does it matter? Yes, it spares tissue, and it also improves functional outcome if you use these mild uncouplers. Just as one target we've used. We've used other compounds that you really don't need to quantify this. You'll see that there's a much less hold here when we administer cyclosporin A, which blocks permeability transition in mitochondria, versus waiting 6 hours to administer. But at 24 hours, it seems to have another effect. And that's something that's baffled us in other compounds, since this is looking at pioglitazone administration. We know pioglitazone gets administered 15 minutes post-injury, very neuroprotective. Pioglitazone, on the other hand, is not active at 1 hour. If you wait 1 hour, it doesn't work, but it seems to restore back at 18 hours. So these are the kinds of questions we're really trying to focus on when we're moving into mitochondrial transplantation. I guarantee you there will be a dose response as somebody in the labs is talking about. We have to know what we're dosing, how much they're dosing. But there's also going to be a therapeutic one, at least in these acute type of injuries. So keep that in mind as you study and move forward with this. We really are trying to nail that down. Same thing with spinal cord injury. In this case, we used we know that oxidative damage alters your mitochondrial complex activity, leading to the loss of function across time, and that we can actually restore this again with this using the cyclolytic carnitine. The intervention Samir, some work from Samir and Sasha's lab, can restore that function, and you can wait as long out to at least 30 hours, maybe even out to an hour, and have some improvement. There is a therapeutic window here again that when it's most effective. It does some targets such as N-acetylcysteine, which neutralizes free radicals, is actually restores walking after spinal cord injury, improves chemomatic analysis, foot stepping after SCI, and most importantly, increases the amount of tissue sparing at the epicenter. That big hole I showed you in the spinal cord, it fills that back in or doesn't allow it to degrade in the first place. So to summarize, we do know mitochondrial dysfunction plays a big role in traumatic brain and spinal cord injury. We know that this oxidative damage also drives a lot of this loss of bioenergetics. It's chicken and egg questions. Hazel will tell you, I'm sure, which comes first, the dysfunction or the mitochondrial damage. Mitochondrial calcium overload is a huge issue. So what about using mitochondrial? This seems like a perfect target, right, to try mitochondrial transplantation, mitochondrial or mitochondrial transfer endogenously. So we tried to standardize some of this. This is work from Samir and Sauce's group, where they took PC12 cells or SY5Y, which are human cells, stained them with Mitotracker red, get a beautiful stain. The issue is, if you filter off everything, the dye leaks, right, and will stain everything you add it to. So when you add it to other cell cultures, such as here, you don't know if you're actually getting a transfer or if you're just getting dye leakage. So that's one of the big issues we ran into right off the bat many years ago. It caused us to move away from that and to actually start expressing RFP or GFP in mitochondria using viruses in these cell culture systems. Again, now that we do it, you know there's not near as much transfer, and especially in 24 hours, because now you're actually looking at how many mitochondria are actually being taken up during this transplant or transfer from this cell culture into this cell culture system. So, being optimistic, we decided to try it in the spinal cord. Okay, let's go ahead and try this in the spinal cord injury. We did 4 different injection points directly into the spinal cord after the injury. Some of the dosing are 100 micrograms in mitochondria, so we do try to dose, keep up with dosing. What we saw was very remarkable. We saw a very nice improvement in mitochondrial respiration, mitochondrial function through various states of respiration, which was a big plus, right? Maybe it's going to work. We decided to go for the internal. We took some of the muscle, took mitochondria from that and injected those. Again, very nice restoration of mitochondrial function. Very easy to see. This is what the injury looks like. This is what happens when you start transplanting different loads of mitochondria. Do note, we did have a slight, we did lose some of the effect when we went to 150 micrograms of mitochondria, so we went back to only doing 50 and 100 in the second study with the mitochondrial muscle transplant. So what about function and tissue sparing? We didn't see any restoration of function. So why? So we went to the histology, and it turns out the mitochondria we injected didn't really get translocated or taken up. They were taken up by resident immune cells, it just seemed to us. So back to the drawing board, right? So next we started looking at hydrogels to see if we could stabilize them and implant them, keep them stable, and release them more slowly over time instead of injecting them in bulk. Again, there's a lot of smears driving most of this work. So we did prove it in cell culture, take isolated mitochondria, RFP level, use the hydrogel, look at the cells. And the one nice thing about using the S5Y and PC12 is we have human mitochondria in a rat cell line. So you can just look at the human mitochondrial DNA, mitochondrial DNA that's there. And sure enough, we show that it's actually being transferred after multiple washes and using these hydrogels. We also been using this in injections, trying to inject it. You see in the intraspinal, you see this big ball. This is your mitochondria. This is TOM20, or this is the mitotransferidine TOM20, and then the overlay, the yellow, which is what you really want to see. And you see that the subdural using the gel, you get a lot better disbursement, which is very exciting. This is blown up here, so you see it really does give you a lot better distribution than trying to use the injection directly. So very hopeful with this. This is going to be another approach, and smears following up on this as we speak, and actually in collaboration with a few people here. So Bailu, who joined the lab about 3 years ago, does beautiful microscope work. You'll see some of that in his talk later today. But we wanted to go in TBI realm. We said, well, let's just try it. You've seen people use an IV injection, so let's give this a shot. Does IV injection work in TBI? We know we have blood-brain barrier breakdown. Seems like it would be an easy target, right? And no, we don't really, compared to the CCI, just a vehicle, we don't see anything but background staining here. And again, we're using 125 micrograms of mitochondria, 250 micrograms, and 500 micrograms. So, and I want to thank Jonathan for this, because Jonathan published his paper, and we got to move away from the dyes completely, and we were able to, he sent us a pair of breeders for his dendromice, so we were able to start this set of studies. So that was the good thing about collaboration in this field, is keep that in mind. If we have any tools you want to use, please contact me or Bailu. Be happy to share anything we can. We did, however, if we isolate platelets from animals that have all their mitochondria labeled, we tried that as well. So we did IV injection of the platelets with these labeled mitochondria, under the assumption maybe they'll go to the target, because we know we have blood-brain barrier breakdown, and disperse the mitochondria there. And again, doesn't seem like that's the picture. We have a lot of platelets showing up, but we don't really see much disbursement of mitochondria in this setting. So, takes us to some of the work. Kazoo's not here today, but Kazoo intrigued us several years ago when he offered up that maybe astrocytes are transferring mitochondria to neurons after stroke. And this is one of the pictures that still intrigues me, labeled. So all the astrocyte mitochondria are labeled green in this picture. So normally it looks like very worm-like, like you'd expect healthy mitochondria to look. After an injury, though, in the penumbra, this is the area that's not going to become the core after the injury. So it's still hanging around as the penumbra of the injury before we impact the brain. They all ball up. Even more intriguing, if you blow out this picture and you really squint sideways, kind of, you can look at this astrocyte labeled blue. Seems to be handing a mitochondria off to the neuron, right? Now, Kazoo will say he showed this, but he didn't really show this in vivo. This has not been shown in vivo yet, that I know of. Astrocytes actually handing them off. But if he was here, we could argue about it. But he left, and he's a crowd. I love Kazoo. So, again, this just shows how different they are. You get these huge worm-like structures versus in the ipsic cortex after you get this very much changing in the pattern. So what's this mean? What are they doing to the mitochondria at this point? You can actually quantify this using automated software. Amaris, Baylor has figured out how to harness some of this power. And again, I mean, you see a very big drop in mitochondrial volume. You see the way their roundness, it's obviously round up, so they do become more spherical. Volume changes, fold changes, using a couple of different approaches. So we know that this happens after injury very rapidly. This is 24 hours post-injury. Are they still functional? So we developed a tool, Baylor driving most of this, to build on some of our magnetic mitochondrial approaches to pull mitochondria down specifically. And we target this, so now we can actually pull down just astrocyte mitochondria after the injury. This is differential. This is all your mitochondria, so you do see about a 50% drop in mitochondrial function, but it's not in the astrocyte. So that's good. So even though they're balled up, which I would think would be a bad sign, they're actually functionally very competent. We also have evidence, so if you isolate synaptic nerve zones, which are basically where your synapse bend and pull the ends of the arm off and you capture those mitochondria. So these synaptic nerve zones, what you see is that you have mitochondria that are coming down from the astrocyte in these synaptic nerve zones. So they are captured in these synaptic nerve zones. This seems to be happening all the time. It seems to be less after CCI, so that's something we're digging into right now to try to figure out more about how to approach that in the future. Finally, we wanted to look more in depth at what's going on with the other populations of mitochondria. We saw the astrocytes look healthy. So what is dysfunctional? So we used some of this technology to actually isolate the neuronal-only mitochondria. Again, this is differential where you see it's about a 50% drop. The somal mitochondria don't seem to really have as much damage, right? And this is new technology, so if you know this field, it's very exciting. We can actually look at multiple populations of mitochondria from the brain. The synaptic mitochondria are getting hammered here, which is not, we've shown that over years, and it's a good target we use for an outcome measure. But we're very excited about this, and it's a ton of work, so just standardizing took us a long time. And again, this is Baylor, and he'll talk more about this shortly today. So to conclude, it looks like mitochondrial function occurs rapidly, progressive over hours, days, weeks, may be reversible. Targeting mitochondrial function to promote recovery of the function after TBI and spinal cord injury. I've shown that to you and proven that to you. You know, we really are excited about trying to leverage astrocyte, transfer from astrocyte or amp it up after using mitochondrial biogenesis. We've got tools. One side of the lab looks at mitochondrial biogenesis and mitophagy, so we're tooled up to do some of this and try to leverage that. You know, and is this transfer contact dependent or independent? That's a question we haven't really answered either, right? Looked like there was maybe contact from the astrocyte to the neuron, but I don't know that I'm convinced yet or not, because we do know we can culture astrocytes. They spit out packages of mitochondria. We can transplant them to cells in other cultures. So lots of interesting things going on. But I do want to, if you don't hear anything else today, note that the mitochondria from different compartments within the neuron even are very different. So we have to be really, I'd be really suspicious of giving a synaptic mitochondria a liver mitochondria as a transfer. And that's just my 2 cents. So with that, thanks.